Hello everyone, my name is Devin Johnson, and today I'll be discussing my research project, the development of a novel 2D tunable hydrogel model to study liver fibrosis. Now, as the title suggests, my research focuses specifically on the human liver. Now, the human liver has so many functions. Some of the most well-known are the detoxification process, the production of bile, and the blood sugar balance that it does for us. However, just like any other organ, it can become diseased over time. Now, the specific disease that I study for my work is liver fibrosis. Liver fibrosis is a form of liver disease in which the liver essentially becomes stiffer over time. Now, this progression can become enhanced, however, due to reasons such as what we eat in our diets, lifestyle habits, and even our own genetics. Since 1980, there has been a significant increase in the mortality rate from liver fibrosis. This health concern causes a need to improve the way that we treat patients with this disease. Now, the goal of my research project was to develop an in vitro model that could be used to better study how cells interact with each other in a healthy liver environment versus a diseased liver environment. This was done with the use of polyacrylamide hydrogels. We made these at varying stiffnesses, specifically about 8 kilopascal and 34 kilopascal, to replicate a healthy and diseased liver, respectively. This was also done in a 2D cell culture model, as this will allow us to better gauge the cell-to-cell interactions. Now, for the materials and methods, there was the hydrogel fabrication process, the immunofluorescence staining and imaging, as well as the QRT PCR process for gene expression. Now, in terms of results and discussion, immunofluorescence imaging was captured for this. What you're seeing here is a control media on the softer substrate versus a 5 nanogram per mil TGF-beta media on a stiffer substrate. TGF-beta is the transforming growth factor beta. This is a protein that essentially induces human dermal fibroblasts, or HDF, cell activation. We then stained for collagen 1 and alpha SMA. These are two proteins that are commonly associated with liver fibrosis. We also quantified these images, and as you can see, stiffness does in fact increase the amount of activation in these cells. We also ran a QRT PCR test, and we specifically focused on the collagen 1A1 and ACTA2 genes. As you can see, not only does TGF-beta induce this activation of the cells, but also the stiffness does in fact significantly increase the amount of gene expression. Now, in terms of future directions, I would like to run this model in a 3D model with the use of organoids, as this will allow us to better gauge how varying cell types interact with each other, as well as how these cell types will engage their varying ECM stiffnesses and how they will respond. Also, these 3D models could potentially be used for patient-specific drug treatments, as you could take a liver biopsy and use the patient's own cells in these models to see what drug treatment would be best for them. Thank you.